The electrochemical potential of a solute is the product of the molar charge of the solute, Q, by the electric potential phi felt by the solute. Phi, the electric potential, itself results from the presence of all charged species in solution. The charge Q of 1 mole of ionic solute of charge number Z is equal to the product of Avogadro's number, the magnitude of the charge of one electron, and the charge number Z, positive for cations and negative for anions. The product of Avogadro's number and the magnitude of the charge of the electron is called the Faraday constant, F. Since there are different ion concentrations inside and outside a cell, there will be different electric potentials phi in and phi out inside and outside the cell. The change in free energy for an ion as it is transported from the outside to the inside of a cell is therefore written as the sum of two terms. The first term is similar to that used for nonpolar solutes and accounts for differences in concentration inside and outside the cell. And the second term is the change in solute electrochemical potential, Z F delta phi. Using this expression for the change in free energy, we can calculate the equilibrium membrane potential delta phi. At equilibrium, the change in free energy is equal to 0 for any constant pressure, constant temperature process. The expression for delta G can then be rearranged to yield an expression for the equilibrium membrane potential. If we consider a typical nerve cell, then the potassium molarity is 30 times larger in the cell than outside the cell, which leads at 37 degrees Celsius to a potential difference of minus 91 millivolts for potassium across the membrane. On the other hand, the sodium molarity is 10 times larger outside the cell than inside the cell, which leads to a potential difference of plus 62 millivolts for sodium across the membrane. This brings us to the last slide of this chapter, therefore the last slide of this course. Here we are now discussing the active transport of ions that takes place through ion pumps. By active transport, we are implying that the transport of ions occurs unnaturally in the direction where both the ion concentration and the ion potential increase. On this slide, this would correspond to the transport of sodium ions from the intracellular space, where sodium ions are in low concentration, to the extracellular space, where they are at higher concentration. This could also correspond to the transport of potassium ions in the opposite direction. How can this happen, since according to the delta G expression used on the previous slide, both the solute concentration term and the electrochemical potential term are positive. Hence, the net free energy change is positive, and these unnatural transport processes should not occur spontaneously. Actually, these processes do not occur spontaneously. They are mediated by the hydrolysis of ATP to ADP, which releases sufficient energy during transport to make it all happen. The process described here is somewhat analogous to that observed for protons when they are pumped across the mitochondrial inner membrane during respiration.